So today we have a very special guest lecturer, Dr. Suzanne Nielsen. She is the editor of your book, and she wrote a few chapters in there, and also the editor of your food analysis lab manual. And she's been teaching food analysis for 35 years, 34? 34. And when I was a postdoc at Purdue, I sat in on all her lectures because I knew I was going to teach this class here. So I can say I learned from the best.
Well, I emulate. I try to emulate the best. I can't try a better class than I do.
No. And I hope you enjoy her. Her area of research expertise is in enzyme. Mostly she studied a plasma enzyme system in milk. And so that's why we invite her over. We picked the enzyme lecture. It seems very appropriate. Although, of course, she's an expert in the other lectures as well. But we chose this based on her research background. With that, let's get started.
Okay, this is the clicker.
Yeah, that's the clicker.
Okay, great. Thanks. Well, it's a real pleasure to be here with you. I hope, can you hear me okay? All right, I put this microphone on to help a little bit. So this must be the TAs, is that right? Okay, all right. Well, it really is a pleasure to be here for many reasons. One, to meet a new set of students, to come back and be with my former postdoc, Pam, and to be, this was my alma mater. I don't remember a class in here when I was a student, but I remember I had biochemistry and statistics in this building. It was a different name at that time, but it was way before you were probably born. But anyway, it's a real pleasure. And as Pam said, this topic is a special topic for me because my research program was on enzymes.
And I forgot to tell them who was your first advisor.
Oh, yes. So some know I saw this earlier this morning, Zeta Vickers. I was only a couple years younger than she is. Actually, I have a lot more gray hair than she does, but I think that's because I was department head for 10 years. But I was her very first graduate student. She was a brand new professor, and so I just got to meet with her for an hour this morning. Yeah, so it's a real pleasure. I did my PhD work actually then in a laboratory in the biochemistry department in Gortner Labs, but the degree was in food science. So great. Well, it's a pleasure to be here. So I'm looking forward to talking with you about enzymes. So let's look at this first slide first and think about, does anybody know what is it about enzymes that of how we get the liquid inside the chocolate-covered cherries? Anybody know? It's an enzyme. It's possible with enzymes. Right? So when they make chocolate-covered cherries, they coat those chocolate-covered cherries with a sucrose, kind of a powdered sucrose with a little bit of a sucrase in it, the enzyme sucrase. And then when that product sits in the grocery store for days at room temperature, that enzyme acts. And just what shows here. So here's the enzyme sucrase. It combines with the substrate, sucrose, right? We get an enzyme substrate complex. It converts to products, and the products, of course, are glucose and fructose, right, and liquid. And that's how we get the liquid inside chocolate-covered cherries. So what you can see is sometimes in food systems, sometimes enzymes are really positive things. It makes things possible like that. When we make cheese, we use rennet. That's an enzyme, right? So very positive things. And then sometimes we're going to see that we don't want those enzymes to be active. And we'll talk about some of the other examples that are shown here. And yes, I left Pam's name on here because honestly, we've worked so closely together on these PowerPoint presentations for your class that I don't remember who developed what part. I know she did the pictures. She certainly did the pictures here. So you have a slide, I think, that looks like this at the beginning, maybe lots of question marks. So let's let you fill in some of those question marks. But let me first ask you, can you think of any reason, a case where we would use enzymes as an indicator of the adequacy of some treatment of, in other words, we use an enzyme as an indicator of something else related to food science? Think of anything? Yes?
I think, don't they test like the activity of a certain enzyme to see if pasteurization?
Exactly. Of what product? Milk, right? Anybody remember what that enzyme is called? alkaline phosphatase, and that's covered at the end of this chapter. Alkaline phosphatase is an enzyme that happens to be naturally present in milk. It's used as an indicator of the adequacy of processing. It's much faster, easier to test for that enzyme than it would be to test for the pathogenic microorganisms that might be present that could really be harmful. So that's why they will test. A rapid test for alkaline phosphatase tells you if you pasteurize the milk, right? So let's go on to the next slide that you have, the slide that shows filling in. Let me let you fill in a few things here. And then I'm going to ask you, we're going to talk about some examples on each one of these, okay? So importance of enzymes in food systems. So as some of you are obviously taking the class, some of you may be just sitting in. I think on the lecture you don't have to take notes frantically on things. But let's talk about food quality. Can you think of any cases where food quality, enzymes are detrimental for the quality of a food product or some characteristic of a food product? Yes?
Pectinases.
Pectinases. And what does that do?
It softens like jarred vegetables.
Okay, and actually pectinases will break down pectin, destabilize pectin. Actually, I thought you were going to say a fruit. What's a fruit that the pectin will break down in with the presence of pectinase? And we get what's called cloud loss. Anybody know what product that is? Orange juice. Orange juice. So pectinase will cause a breakdown of the pectin, destabilizes it, and on that picture that we had just a bit ago here, that orange juice, this orange or other citrus fruits will naturally contain pectinase. It destabilizes the pectin and it causes it to fall out of solution. We get a kind of a clear juice, not very appetizing. Perfectly safe, but not very appetizing. So that would be one, pectinase breakdown. Can you think of another one? What is the type of enzyme that causes hydrolytic rancidity of fats? Anybody know what that's called? Lipases, right? Lipases can act and cause hydrolytic rancidity of fats. What would happen if I had some starch, a starch paste that was supposed to be thick, right, as a starch paste, but if there's a little bit of amylase in that starch paste, what's going to happen? It's going to lose all the viscosity, right? So there's a case where we don't want that loss of viscosity, so there's a case where amylase activity may be undesirable, okay? So those are a few related to food quality. We mentioned already one on the processing tool. Rennet is used as a processing aid for making cheese, right? Okay. Let's think of another one. Let's use your pectinase example. What if I wanted clear apple juice? Then what do? I make sure the pectinase is active, right? If I want cloudy apple juice, I heat and activate that enzyme quickly, so I get the cloudy apple juice. If I want clear apple juice, I make sure that enzyme acts so I can get the breakdown of the pectin and I can get the clear apple juice. Okay? Waste management. Sometimes we use proteases or lipases to break down the proteins or fat in a waste stream coming from a processing facility, so we don't have as much organic matter left for decomposition. Measuring activity. We talked about one of those already. We talked about alkaline phosphatase, the enzyme that's used as an indicator of adequacy of pasteurization of milk, right? Do you know what an enzyme is that's commonly used as an indicator of adequacy of blanching? Drew, that might have been the one you were thinking of also with blanching. Do you know what that is? Peroxidase. Peroxidase, or some people will use lipoxygenase, but peroxidase is commonly used as an adequacy of blanching vegetables, right? Because if we don't inactivate, if we don't blanch, we're going to end up with vegetables once they come out of the freezer that don't have the same structure, the same substance that we want. Okay, and sometimes we can use enzymes to quantify other food constituents. So, anybody want to guess that the assay for glucose? That's an enzyme assay. We're going to see that one. Also, the assay for starch, what would you guess, what enzyme might you use to assay for starch? What breaks down starch? Amylases, right? And we're going to see that what we do to measure, the best way to measure starch is to break that starch down to glucose using amylases. And then we use another enzyme system to measure the amount of glucose that we have. We'll see that assay a little bit later. Sample preparation. So did you already cover carbohydrate analysis? Right? And fiber, did you cover fiber analysis? Right? Do you remember in that assay for fiber analysis, what enzymes are used? What do we use to break down the proteins? Proteases, right? Two amino acids, and they can get, now they're not soluble in high concentration of alcohol. And what do we use to break down the starch? Amylases, right? To break it down to glucose, and then again, they are soluble in high concentration of ethanol. It's also used in sample preparation for thiamine assay. If you're going to do a thiamine assay, usually there's a treatment with enzymes there. And then the last thing that's shown here is isolation and characterization, where we're really concerned about the enzyme kinetics. In a research lab, this is what my students used to do a lot of, isolation and characterization of proteins or enzymes, enzyme inhibitors, and we're especially concerned about the kinetics there. So there's lots of reasons it's appropriate to talk about enzymes related to food analysis. Okay, so let's go on, and what we need to do is, in this first section, is talk a little bit, we're moving toward a bit on the enzyme kinetics. Now don't think back to all the chemistry and biochemistry you've had and say, I thought I was done with that. But we need to talk about it just enough so we know how to control the enzyme reactions when we use those enzyme assays in food analysis. So here's an enzyme, a simplified version of an enzyme catalyzed reaction, right? You probably saw this before where we have an enzyme and a substrate. We get an enzyme substrate complex, and we get the enzyme regenerated, of course, and then we get the product. We measure the amount of product, and that can tell us either the amount of the substrate that we had, or it might tell us the amount of the enzyme that we have, depends on what we're after. Okay? These are just rate constants up here. You can see here we've got to have two different events. We've got to have that stereochemical binding of the enzyme and the substrate, right? And we need this chemical conversion to generate the product. So that's what's happening in our reaction here with sucrase. This is just reminding us that any chemical reaction, there's energy that's required. If we have an enzyme involved, it serves as a catalyst in that reaction, and it takes much less energy for that reaction to occur, converting the reactants to the products, if we have an enzyme involved there. So we reduce the activation energy. Okay, let's move on, and we have the same equation over here. We have the same equation of the enzyme catalyzed reaction, and what we see here, starting down here, we have just the enzyme and the substrate are being complexed here. As we move up here, we have product starting to be generated, right? So we're going to see the importance of this part of the slope right here in an enzyme catalyzed reaction. The slope, as it says here, the slope is the initial velocity. That's the V naught, is the initial velocity. Okay? So that same diagram is shown right here. In this steady state, here's where we can measure the initial velocity, it's the slope. And as we've got three terms that are defined here, the initial velocity, maximum velocity, and Michaelis constant. This is the part that relates to kind of the enzyme kinetics, but we're going to see that that relates to what we have to control when we do the assay. So the initial velocity, as we just said, it's the slope of this portion of that curve where we generate the product. The maximum velocity is up here, is clear up here. And Michaelis constant, this term Km, is a term that's used to describe the binding of, as it says here, the binding of the enzyme and the substrate. So the tighter that binding, the lower the Km value is going to be. Km, Michaelis constant, is an important characteristic for each type of enzyme catalyzed reaction. right. So what we have is those same three terms as described here, and now we have this figure that is called the Michaelis-Menten diagram. Guess we would saw a plot of the Michaelis-Menten equation. We're going to see that equation in a minute. Let's look at the last term that's described here, Michaelis constant. The substrate concentration at half the maximum velocity. That's what's shown right here. So here's the velocity, here's maximum velocity, here's half maximum velocity, and if you come down here, the Km is the substrate concentration at half the maximum velocity. So that's the definition of the Km, and it's important because it tells us something about the binding of the enzyme and the substrate. Okay? Now, we're going to talk in just a minute about what it means if you're down in this part of the curve, versus, of the plot, versus in this part, the high concentration of the substrate. Okay? So here's the RKM value, and here's the Vmax. The reason we have to talk about these is because they are affected by all the kinds of things that you have to control when you do an enzyme catalyzed reaction. When you do an enzyme assay, and I understand in your lab, I think you're not going to do an assay, but in the class that you have to take next semester, you're going to do some of this. You're going to do an enzyme assay, and you'll need to control all of these factors because you can see that they affect the Km, Michaelis constant, and they affect the Vmax, the maximum velocity. So what I'm going to describe a little bit later is an assay. These are the set of instructions that come from a test kit. Okay, long set of instructions come from a test kit for an enzyme. It's an enzyme test kit that you can buy. And the point is, in that assay, they have taken care of everything. They've supplied you the reagents, they've told you the time, the temperature, the pH, everything is controlled because when we do that assay, we have to control all of those factors because they affect the Km and the Vmax value. Okay? And you can see this Vmax, it's affected by many of the same kinds of things that control the Km value. So when you do that assay, you're doing it at a certain pH, at a certain temperature, you're controlling all of these factors, and therefore you're able to control the Km and the Vmax value. Okay. Now, in an enzyme catalyzed reaction, if what we want to measure is the substrate, if we want to measure something like starch, then what we want to do is we want to use a substrate concentration much less than the Km value. Okay? Versus if what we want to measure is enzyme activity, like if I want to measure amylase, we want to be at a substrate concentration much greater than the Km. So let's go back just a minute and remind us what that means. That means in one case we want to be down in here, in the other case we want to be way up in here. Okay? And you can control that with your assay, but it depends on what you want to measure. Okay, so here's our Michaelis-Menten plot. Here's the Michaelis-Menten equation. I think, as I understand from Pam, you don't have to memorize that equation, right, in any way. My students, when I say something like that, they all go, oh, okay, good.
They will get it in chemical reaction.
They will get it next semester, right? Next semester. But we're going to talk about it just enough to get to the point of the important points here. So here's the same plot. This is the equation, but many times we would rather work with a straight line, wouldn't we, than to work with a curve. So what we do is we use the inverse of that equation. And here what we have is called the Lineweaver-Burke equation. This is a Lineweaver-Burke plot. It's a straight line plot as compared to the Michaelis-Menten equation and the plot that is a curve. So instead of the V, the initial velocity over here that we saw in the Michaelis-Menten, we have 1 over V naught. And instead of 1 over just the substrate concentration down here on the x-axis, like we had in the Michaelis-Menten equation, we have 1 over the substrate concentration, right? So everything is inverted. And this way, if we were to do the assays and we generate a plot something like this, if I know the maximum velocity, and I know the slope of this line, I can calculate the Km. That's how you do it. If you needed to get that value, this is how you do it. Is that what they're going to do in class? Next semester. Next semester you're going to do that. So we change over from the line from the Michaelis-Menten equation to this one. Here's the Michaelis-Menten type of a plot that we have over here. We use a Lineweaver-Burke and we get that Km value if we wanted to know that. The important point I think for your purposes is to know what kinds of things you have to control to be able to control that Km value. All of those factors you have to control. All right, so let's move on now and look at those factors we have to control just to remind us what would be the effect when we change those things. So if we change the enzyme concentration, here's at least the theoretical value. You can imagine if we increase the enzyme concentration, what should happen on the velocity of the reaction? It should go faster, right? That's what we would expect, okay? Now, this plot shows 4 different concentrations of the enzyme. So increasing from 1 to 4. The dashed line are the experimental ones, and the solid lines are the kind of the theoretical, it's the slope of that line, okay? Now, imagine that this is the time point where I measure that assay. Time A right here is where I'm measuring that. So you can see if I have the enzyme concentration at 1, and if I measure the activity at A right here, here's how much product I'm going to have generated, right? As compared to if I use concentration of enzyme at 3, here's the concentration of the product that I'm going to have. So we have to control that. We control the pH. We control the time. Do we let the reaction go to completion, or do we stop it? Is it a 10-minute assay? You know, if it says it's a 10-minute assay, you've got to make sure that enzyme reaction stops at 10 minutes, right? Okay, let's go on to temperature. Another factor we've got to control. When I do this assay with this test kit, it tells me exactly what temperature I need to use. It tells me what pH to use. This shows 3 different plots here. Number 2 is the final result, but number 1 is increasing the temperature. So if I increase the temperature, I should theoretically get more product generated, a faster velocity to that reaction, right? Okay, increase the temperature. But enzymes are proteins. What happens if we get to too high a temperature with a protein? Denaturation, right? And that's what's happening with number 3. We're getting denaturation. So yes, we want to increase the temperature to a certain point, but then, you know, so maybe this is optimum for that particular enzyme. What do you suppose the optimum temperature for the enzymes in your body is? Probably 98 degrees Fahrenheit, you know, 37 degrees centigrade, right? So some assays we do at 25 degrees centigrade, some at 37, some at 45 degrees centigrade. It depends on the enzyme. Whatever's the optimum for that particular enzyme. What about pH? Okay, so for each enzyme, we'll have a different optimum pH. This plot reminds us of the pH stability versus the optimum pH. Again, these are enzymes, so we may destabilize the enzymes at a certain pH, but we want a pH that's necessary, and the optimum would be necessary for ionization so that we can enhance that enzyme substrate complex there. And also so we can generate the amount of product. So we've got to be at the right pH for that. So another example, an example for that is trypsin. The enzyme trypsin is very specific. It cleaves at just 2 different residues of amino acids. Trypsin, I remember working with a lot of trypsin when I was a graduate student up in Gortner Labs, and I'd make up a solution of trypsin, but I had to make up that solution in pH 3, like it was a dilute hydrochloric acid solution, because anybody know what the optimum pH for trypsin is?
It's over there.
It's right there. Oh, it's right there. Oh, yes, it's right there. Thanks. 7 to 8. Okay, and not surprising, that's the pH in your intestines, right? Right, in your intestines. So That's our optimum pH. At pH 8, if I make up a solution of trypsin, just plain solution by itself with no substrate present, trypsin will chew itself up. But if I hold it at pH 3, it doesn't chew itself up. Now when I want to start the reaction, I bump the pH up to 7 or 8, and I make sure there's a substrate for the enzyme to act on. Okay? Yes. So there's our optimum one. What about pepsin? What's the optimum pH for pepsin?
2.
About 2 to 3, right? Good thing, it's in our stomach. Okay? Okay. Now, sometimes we have to add activators to the assay. We have the enzyme and the substrate, but we have to add an activator. Not for all enzymes, and it describes here what those might be. Maybe a prosthetic group, a non-protein portion, sometimes a coenzyme. When I would do those assays with trypsin, I had to make sure there was a little bit of chloride ions present because that was a bit of an activator for the trypsin. Okay. Inhibitors. Sometimes we want to measure inhibitors, the amount of inhibitors. Sometimes we need to make sure there aren't inhibitors present that would prevent the reaction from going to completion. This describes different types of inhibitors, and Pam, you'll have to tell them, I don't know if you expect them to know all the different kinds and the details of the, I always tell my students,
They take that in food chemistry, so they
yeah,
should
so they
know
know
this
something
already.
about different types. There's different types of inhibitors. You know, an example of a food example related to a trypsin inhibitor, an inhibitor is trypsin inhibitor. Trypsin inhibitor is naturally present in soybeans, raw soybeans. So soybeans, when they're processed, they will typically roast those soybeans in part to inactivate the trypsin inhibitors. In fact, when I worked in biochemistry for my PhD work up in Gortner Labs, my professor was an expert on trypsin inhibitors. That's part of what he did. And he would do these rat studies, and then actually one case, a human study on the effect of trypsin inhibitors. Did it really shut down the trypsin in your digestive system? Okay? But, so let's imagine that I've got these soybeans. The soybeans were roasted, but maybe they weren't roasted. I have some that I think were roasted adequately, and some that were not roasted adequately. I want to measure the amount of trypsin inhibitor that's present in both of those batches of soybeans. I've got an enzyme, a substrate, and then I'm going to get a product generated. And what I want to test is the amount or the activity of the amount of the inhibitor that's present. So if I have any inhibitor present, what is it going to do to the amount of product that gets generated? Reduce it, right? So for that assay, what you do is you use a substrate. It has to be something that trypsin can act on. Typically to test that, they use this little polypeptide called BAPNA, benzoyl arginine pyranidroyl, and it's a little chemical you can buy. And you react it with trypsin, right? And now I'm going to get a product generated. In this case, it's a yellow compound that gets generated. I measure the amount of yellow color. If I have trypsin inhibitor present, I will get less color present, right? And if I've got more trypsin inhibitor, maybe because I didn't inactivate those soybeans totally by heat treatment, that's going to have an effect on the amount of product that I have, right? So we can use an enzyme reaction to measure the amount of an inhibitor that's present, okay? Now, how do we do that? In the case of the example I just said, we would measure absorbance at a particular wavelength. I can't remember which wavelength, but we used absorbance spectroscopy to do that. Simple, you just use a spectrophotometer, right? But there are some other ways to do it. In fact, the bottom one is an interesting example, viscosity. So let's say I have some wheat flour that I want the starch from the wheat flour to make a starch paste. And I am concerned that there may be some amylase activity. Sometimes if grain gets warm and moist, it thinks it's supposed to start to germinate, and it will start to produce enzymes, amylases, proteases, right? And as we already said, what's going to happen if I have some amylase produced in my wheat before I process it into flour? What will happen when I make a starch paste? It's going to lose viscosity. And that's exactly how they test it. They measure viscosity. It's called a falling number test. You make a starch paste and you have a plunger, and you measure the amount of time it takes for that plunger to pass through this starch paste. So there's a case where what we measure is viscosity. And you can see there's other ones in between. Probably the most common is an absorbance spectroscopy. Okay. One other important concept before we get to some of the examples of enzymes again is a coupled reaction. We talk about this because many of the assays that are relevant to food analysis, the enzyme assays are coupled reactions. That means it's a two-step reaction, right? We have a measuring reaction and we have an indicator reaction. What we measure is some combination of these products that are being generated, and that goes to our spectrophotometer to measure the amount of absorbance that we have. Now, a couple tricks with that. Well, here, first one example. Here's the most common assay for glucose. It's an enzyme catalyzed reaction. It's a coupled reaction. So the first one is glucose reacting with glucose oxidase. Oxygen, we get hydrogen peroxide generated and gluconodelta-lactone. That peroxidase, that peroxide then is reacted here with another enzyme, peroxidase, and we get a colored dye that's generated. Most common method for measuring glucose. Enzyme catalyzed reaction, very specific, right? Okay. It's a coupled reaction, two steps. In this case, all we have to measure is the amount of the oxidized dye that we have down here. Okay. A couple of cautions with regard to those coupled reactions. You can imagine that if you don't have the two enzymes you're using don't have the same optimum pH, it's not going to work very well. If one of them has an optimum pH of 3 and another one has an optimum pH of 7, it's not going to work. Right? It's difficult to adjust that in between. So if, in fact, this example I'm going to talk about here next, this is a coupled reaction, but both of those enzymes have a similar optimum pH. So it works. If it doesn't, if they don't have a similar pH here, you probably got to rethink that. The second point is here that this enzyme 2, this second one, cannot be rate limiting. This has to really be going faster to make sure everything is pulling through to finally generate the final product. Okay, not surprising. And we cover these coupled reactions because many of the assays that we do in food analysis are coupled reactions. Here's the one that I wanted to, another coupled reaction that is the basis of this test kit. I was kind of short on space in my luggage, so I didn't actually bring the test kit. It's a little box, but it contains everything you need. It included these instructions. I brought those. So this is a little overwhelming. I make my student, when I was teaching lab, they would have to read through this. You know, I would give them the Cliff's Notes versions of it also. But it's difficult to read through, but it tells you everything you need to know to be able to do that assay. How to prepare your sample, how to dilute your sample, how to use each one of the reagents that they give you to do that assay. And that's all these reagents. We would use it for measuring ethanol content of honey. Believe it or not, honey contains small amounts of ethanol, and we could use these test kits to do that. You notice in this particular case, we're using two different enzymes, alcohol dehydrogenase and aldehyde dehydrogenase. Okay? So to do that assay, what you would actually do is we would prepare our sample of honey, we would clean up the sample, dilute it to the right concentration, close to the concentration that would work, and we would combine it with all the reagents except this first enzyme. You measure the absorbance, and then you add the first enzyme, the whole reaction series starts, and what you do at the end is measure the absorbance again. The first one was kind of the blank absorbance. The second one, we're measuring the amount of NADH that gets generated. In this particular case, we get 1 mole of NADH produced for each 1 mole of ethanol. Now that's how this particular one worked, but these coupled reactions, it's just a matter of how it's set up and how it works. It was interesting, in this case, we didn't start the reaction. You don't start the reaction with this enzyme. You actually have this enzyme present already, and you add that first enzyme to start the reaction. All right, now we're going to come back and just remind ourselves, if what we want to measure is the amount of the substrate, like starch, like glucose, those would be substrates, then a reminder, we want to use a substrate concentration that's much less than that Km value. Remember, the Km was the substrate concentration at half maximum velocity. So to measure these kinds of things, we need to be in this position typically. And if what we want to measure is the amount of an enzyme, if what I want to measure is amylase, or sucrase, or alkaline phosphatase, or pectinase, then I need to be at a substrate concentration much greater than the Km value. Again, if you buy a test kit, they've got that all figured out, and they tell you how to do it. But this is what they're trying to follow these same principles.
May I add something,
Yes.
because it's going to be part of what they will be examined.
Yes, that's good. That's good.
So if you go back one slide, Susan.
Yes.
So this is the part of the curve, if you remember, where you have linear curve with the products being produced. So if you want to measure substrate, you want to make sure you're in the linear range of the reaction, so that the production of the product, you can link it in a linear equation and get the concentration. And then the other one is when you need the enzyme, you want to make sure that the enzyme is saturated with the substrate.
So you're
So
clear out here.
You're up there, yeah. So when
You're
you
up
have
here.
maximum velocity, then the reaction is first order with respect to the enzyme. So if you change the enzyme concentration, then you will see a change in the reaction. So that's just because you're going to be tested.
No, that's good. That's exactly. I asked her what you were going to be tested over. So,
I forgot to tell you
that's
that.
good.
Yeah.
So remember back to those early plots, those early figures we had. It mentioned the first order and zero order reactions.
Yeah.
Obviously, you want to go back and look at that in
Yeah.
more detail.
Yeah.
Yes. Okay.
Yeah.
Good point. All right. We're almost done here. This slide just reminds us that sometimes we use biosensors. For example, you can use a glucose electrode it's based on glucose oxidase and the assay for doing glucose, but we can use these as biosensors. There's some increasing use of these. In fact, the new author of this chapter for the next edition has added some more information on this topic of biosensors. And the last thing is, again, reminding us all the application. When would we use these enzyme catalyzed reactions in food analysis? So a reminder, alkaline phosphatase, rennet, those are commonly used in the dairy world. So for example, I can remember for rennet, have any of you worked in a dairy processing facility where they make cheese? Anybody seen that? And so there's the big jugs of Chymax, or it's the genetically engineered version of the rennet. Right? And sometimes over time that will be less active. So I've seen cases where they've got a big vat of milk ready to make the cheese, and if that rennet solution has been sitting around a while, they test the activity of that enzyme solution to make sure they're adding the right amount of that enzyme to make the cheese. Right? The alpha amylase, we have already talked about that. It's called a falling number test, is what's commonly used for measuring alpha amylase, because we said if it starts to get warm and moist, it thinks it's supposed to start to germinate. It produces amylases, and then you end up with amylase in your flour. So a common quality test for something like all-purpose flour would be a test for alpha amylase activity, doing a falling number test. Pectinethylesterase, for the reasons we talked about here, the cloud loss. Either you want to fully inactivate it quickly, or you want to make sure it is active to break down the pectin. Sometimes we live with the amount of heat-stable pectinases that are still present after processing. And for vegetables, probably the most common test for blanching, adequacy of blanching, is testing peroxidase. You'll see in the book it also mentions that you can test lipoxygenase as another one. Can you think of it? Is there another example, Pam, you wanted to give
No.
besides enzymes? These uses? So my guess is you're doing these projects for your, for class, right? And I'm guessing there'll be a few of you at least who will have quality tests that will show up in your products that are related to enzymes. Many times if you've got a dairy product, one of the tests that may be done for adequacy of pasteurization is that alkaline phosphatase test, right? If it's when my students do those projects and if they've got wheat flour, for example, typically there will be a test somewhere in there for alpha amylase activity. If they're assigned orange juice, sometimes there's a test for pectinoglycerase, right? And anyone who has frozen vegetables assigned as their product, it usually shows up as the peroxidase. Okay, I've talked on and on, and we've got a few minutes. Any questions? So what's the hardest part? Let's help summarize that whole part about the kinetics, right? And I think from what I understand of you is similar to what I tell my students. The big part about the kinetics portion of this chapter is understanding why we have to control all these variables when you do these enzyme catalyzed reactions. You've got to control the enzyme concentration, the pH, the temperature, all of those factors, because you can see how all of those affect the Km. The Km is that binding. It's an indicator of the binding of the enzyme and the substrate, right? And it controls the velocity, the maximum velocity, so that's why when we use this kind of a test kit, all of those factors have been taken into account, and I don't have to worry about it. But somebody had to figure that all out in order to develop these kinds of test kits. So, yeah. So I understand you did the phenol sulfuric acid assay. Some of you at least did it today, right? No injuries, right? I can only remember early in my career I had one student who got a little carried away with the sulfuric acid and had a little trouble. But
Yeah, we had the student splash on her face, and she still has a scar. Yeah.
yeah.
So we're scaring the Wednesday group now.
No, it happened today? It happened just today or years
No,
ago?
a couple of years ago.
Yeah. You know, it's an easy one to burn a hole in your jeans or something, so just be careful with that. But it's a great assay. It's really a very, very useful assay. I mean, I used to use that in graduate school to estimate the amount of carbohydrate that was attached to a glycoprotein.
Yes, we do that a lot.
Is that what you do that? Yes. So it's a great assay.
Mhm.
But you've got to be consistent, right? In pressing, do you have a pump?
We
to
have a
deliver-
repipette.
A repipette to deliver the amount of sulfuric acid, right? And you've got to do it fairly forcefully
Yeah.
to mix it as you add that, and you vortex it right away. If you let it just dribble down the side of the tube, it
Messes up the, yeah.
it will mess up. The precision will be terrible. I've had graduate students who thought they would give up on that assay because they couldn't get good precision. And it was just that they weren't getting enough heat generated in that by forcing the sulfuric acid into that tube quickly to do that.
Yeah. You can see I got my training from Suzanne. That's what I told him in the lab.
It's true. One of Dr. Cousin's students, she was going to give up on that assay, but they were letting it carefully dribble down the side of the tube because it was sulfuric acid.
Yeah.
Of course, but it's not the right thing to do for that particular assay. So, yeah. Well, anyway, good luck to all of you in your projects that you do. So, we've got just a couple minutes. Can I quickly hear what kind of products you're doing what you're assigned for your products? Anybody?
You can volunteer.
Cheddar cheese. Oh, good.
Protein bar.
Which one?
Protein bar.
Protein bar. Ah, okay. What else?
Semi-sweet chocolate.
Semi-sweet chocolate. Ah. What else? Anything else?
Peanut butter.
Peanut butter. Ah, that's a good one.
Blue cheese.
Blue cheese. Yes.
High protein beverage.
Ah, okay. Oh, one of my students did that one last year.
And guess
Yes.
who was the industry?
Yes, yes. This is her industry contact.
I was at Cargill.
She was
so
at
what's-
Cargill. Yes, yes. What else? What other products? Any other products? Tofu, oh, interesting. Yeah. So, my students do the same sort of thing. I'm guessing Pam may have gotten that idea from.
Actually, Lloyd was doing this before
okay,
I came.
yes.
I just elaborated a little bit.
Yes, yeah, and I think, yeah.
Oh, Lloyd maybe got it from you.
I think so, but it doesn't matter. But anyway, it's a great project. I think it connects you with somebody, some expert in that area. Yes.
So how big is this food analysis teaching?
For me,
It seems like you guys share a lot.
we do. I was saying that this morning, it really is a team effort. It started out, I got talked into doing that book in a way because there wasn't a book, there wasn't a textbook. I got my first, when I went to Purdue, I really wanted to teach, I told the group this morning, I wanted to teach food chemistry. They didn't need anybody to teach food chemistry, but they needed somebody to teach food analysis. There wasn't a textbook that was really suitable for students. It was more of a reference book that was available. And I was using the class notes that actually came from Minnesota because I didn't take that class when I was
See, it's a full circle.
Yeah, the original thing came from Minnesota. But I started teaching it and I got a request to review a proposal for a book. Somebody else was going to write a food analysis book. I reviewed it, I was critical of it because I didn't think it was going to cover what was needed for teaching it. So they came back and said, well, why didn't you write it? and I needed a book. That's the only reason we got, but the people I asked to do the chapters are people who teach food analysis, mostly, who teach food analysis. So it's lots of people across the US who teach food analysis and a couple beyond that. So it is a team effort. It has been, and we share a lot of the resources, the teaching materials. I put on a website, there's lots of the teaching materials that people have contributed. So it is, I think unlike probably any other part of food science right now, there's this cohort of people, and not just in the US, but I get requests from other parts of the world. And it's a team effort, I think is what people do. Because none of us has enough expertise to teach everything, to write chapters or write portions on those topics. So it has been, there's more sharing, I think, of that material than probably for any other part of food science. So it works. And we do it because for the students. I think that's why we all do it.
How long did it take to put together that first book?
Oh, it was a long time. I was doing it for years. I don't know what I thought I was doing, but it was not easy. Every time I do it, I go, and somebody says, so you're going to do another one soon? And I go, oh, we don't talk about it. But you know who's taking over next time. This is my last time.
Oh,
She is taking it over. So
I was talked into it.
So the fifth edition, it should be out in early 2017. I'm working on page proofs right now, and but this is it. This is my
I'm
last
gonna
time.
help next time.
I'm going to help, not much. So Pam is taking it over. So I'm entrusting it to her.
Well, we're gonna call it the Nielsen's for the
Well, who knows what it will be called.
like
but-
the Fanamas with chemistry. It should be the Nielsen's.
Yeah, well,
And my name in very small font.
It's been a team effort. I'm glad you asked that question. Really, it's a great group of people that I get to work with who they're glad to contribute now because they all need a book.
So do you guys meet physically?
No, we never. And maybe at IFT once in a while. But we don't physically meet. But it's there's this camaraderie, I guess. And for new faculty who step into needing to teach it, it's we hope it helps make it easier because it's a lot of work.
Yeah, because you're offering the PowerPoint, you're offering all the teaching material.
Yeah, the teaching materials. So it's it's work. But anyway, it's been a pleasure. I know you need to go, but it's been a pleasure being here with you today. I really wish you all the very best.
Good, thank you.